Good afternoon, everybody, and welcome to the Strategic Cereal Results Farm for Results webinar for 2022. This particular one will be all about improving establishment and yield, and the things we're looking into, the things that we've found so far. And as you know, the Strategic Farm series is a 6-year program, so this is a look-see into the progress and results that we have going so far. So can we go on to the bit of housekeeping, please, Christian? So yeah, housekeeping wise, you are all muted. We can't see any of you, which may be a good thing. Hopefully you can hear and see all of us. In terms of questions, we've got a chat box you'll see on your right hand side where you've got a small line saying questions. Open that up, and as people here are talking about different things, think of the question, pop it into the chat box, and then it's saved, and then we'll have a roundup of questions at the end, and we'll come back to our various speakers to ask some of those questions. We are planned to run from 12:30 through to about 2 p.m. In a kind of loose agenda. We might finish slightly earlier, but that gives us room for some chat and some extra questions as well. And basis and Naroto points have been applied for. If you want to put your basis and Naroto point in the chat box, then that would work easily well. And yes, and this also will be recorded and put onto our YouTube channel in due course. So that's the housekeeping done. So oh yeah, the next thing was well reminded, Christian. So today is 9th of November. So you're here and live on the Improving Establishment and Yield. Next week, 16th of November, Farming and the Environment with Anna Reynolds. 23rd of November, the week after, Tailoring Crop Inputs, very relevant just now, with Fiona. And then finally on the 30th of November, the future of the sustainable arable business with the infamous Isabel Ames from York. So make a note in your diary. We'll try and get this slide up at the end of this webinar as well, in case you need any reminding. But those are the headline events happening for the strategic farmers. So without further ado, I'd just like to start at the south first. Really, we'll start with Callum, and we'll go to David Miller, then David Aglen, then Fiona. Just to introduce yourselves and just what your connection is with the Strategic Farm Programme.
Hi all, so I'm Callum Scottson, so I work for NIAB. I'm based down at the Sutton Scotland Centre in Hampshire, and I've been managing the Strategic Farm South from NIAB's end, and that's for all 4 of the packages. All 4 are sort of soils related, and in terms of the other work that I do at NIAB, I essentially manage a lot of the portfolio in terms of projects for soils and farming systems work in this part of the country. And my background is that I've got a PhD in effectively soil science and how plant roots interact with soil. Yeah.
Great stuff. Thank you, Callum. And David Miller, to quickly introduce yourself.
Yeah, I'm David Miller, manager of Wheat Sheaf Farming, and obviously the Strategic Farm for the South. Yeah, the background of what we're doing on the farm really is we've been going down what we now know as a regenerative route since about 2010. So very pleased to have the HDB and NIAB on board to try and understand what we're actually doing. Thank you.
Good, sir. Okay. Super job, Fiona.
Hi, Fiona Burnett from Scotland's Rural College. I'm an applied plant pathologist, and we've been working with David Aglen on the Strategic Farm in Scotland, along with a team of colleagues. But my interests cover regenerative agriculture, integrated management, and more sustainable practices. And what David's doing is, really pushing the edges of what farmers are doing in Scotland.
Yeah, great stuff. David Aglen, do you want to introduce yourself?
Good afternoon, David Aglen, Farms Manager at Balbirnie Home Farms up here in Fife. We are the Strategic Farm for Scotland, as has already been mentioned. and we are trying to farm in a more sustainable manner, maximising the use of resources we have within the land and the business as it is now. And we're going in a more regenerative direction, and keen to use the strategic farm as a way to try and research some of the things we're trying and get some answers, and hopefully help others make decisions as well in their own businesses.
Yep. Okay. Super job, David. And just joined us now, and joined us literally and with the strategic farm, David Blacker. David, are you able to introduce yourself and your business?
Yeah, can anyone hear me?
Yeah, that's good.
So I'm David Blacker. Just started Strategic Farm North, ex-monitor farmer, farmer under contract from Shipton in the Vale of York, predominantly heavy clay loam soils. We've just got going with Strategic Farm this year, and obviously year one, so we haven't got any results yet. Here to lay the scene of what we're going to do.
Yeah, great stuff. And just to introduce myself, Harry Henderson, Technical Knowledge Exchange for HDB. So what we do is start in the south of the country, and we'll just hear what work Callum's been doing at Wheat Sheaf Farming. And over to you, Callum.
Let's get back to that. Super. Hang on a second. There we are. Right. Hopefully that's displaying okay for everyone. I'm sure someone will let me know if it isn't. So as I say, I'm Callum Scotson, managing the Strategic Cereal Farm South from Knives End. What I'm going to be talking about today is the establishment and route development project that we've got going on. So it's one of 4 different trials that we've got on site, where we're looking at various different bits and pieces. All of them tie in to a greater or lesser extent with sort of soil science, monitoring soil health, and different aspects of that, and how it relates to the management on farm. So I'll just give a bit of broader context of what we've got going on farm. So these 4 trials. So the first trial that we have is a cover crops and water quality trial. So we've got sort of a field of winter wheat, and we're comparing that against some strips of different cover crops effectively, and looking at how those interact in terms of soil health, water quality. We've also got another trial which looks at 4 different management styles, so 3 of which David's managed for a long time under his system that uses sort of principles of regen ag, min-till, progressing to no-till, that sort of thing, comparing them to another conventionally managed field. We've got the trial that I'm going to be talking about today, which is that establishment and early crop growth trial. So looking at 2 products applied at drilling, how those affect early crop growth and establishment. And then we've got a broader sort of farm scale monitoring trial, looking at how to baseline soil health. And we'll be covering some of those other trials in various other webinars as well. So as I say, that's the one we're going to be talking about today. So to provide a bit of background as to how this trial is set up. So it's located within this field. In terms of the trials that we've got on David's farm, it's one of the more sort of simple to explain in that it's a fairly conventional tramline trial effectively. So we've got replicated tramlines where we've applied either nothing, it's an untreated control, so it's completely untreated seed, no seed treatment or anything. And these are indicated on that little plan of the field down in the bottom corner there. We've also then got an eco-worm application. So again, it's applied at drilling. So we've got eco-worm tramlines. We've got tramlines to which LCBF boost was applied, and we've got tramlines to which both were applied as a combination treatment. Effectively, eco-worm is a vermicompost, so it's effectively the product that you get out of worms decomposing organic material. And then LCBF boost is a molasses-based starter for and it's looking at effectively how these 2 products influence that early crop growth and that sort of root development. So in order to assess that, we look at a number of different features. So we're comparing the soil health between the tramlines. To some extent, this is to make sure that the soil structure, for example, is consistent between tramlines. And in other instances, we're actually looking to see where those differences develop. So it's important to caveat this trial in particular, actually, that this is obviously the first year of this project. So more data, the data will develop basically as the project progresses, and we'll likely get those changes with time developing. But if we do see changes, that will be where it is. It's as it progresses. It is also important to state that because again of the nature of the trial, we are looking at a soil type, a field in a particular year. So there's no guarantee that these same trends that we see here will take place in other soil types and in other years. But we also look at other features. So we look at early emergence, crop development vigor, monitor that root system development, as I say. And we've done a few other bits and pieces in terms of soil microbiology. So quantifying microbial activity in the rhizosphere, sort of before and at the end of winter, and also looking at arbuscular mycorrhizal fungi symbiosis within that rhizosphere, within those wheat plant roots at the onset of. So this is something I'll run through very quickly. It's a little bit of a sort of technical aside, really. This is just how we identified the region of the field to run the trial in. So it's a method called spatial clustering. So the idea is that it's a method that for use in this application has been worked on by my colleague David Clark at NIAB, and effectively it enables you to look at, in this instance, wheat yields from previous years, look at how those overlay, how they cluster together to find regions in which there's a bit of consistency between years. Basically enables you to avoid corners of the field for a trial such as this, where you might get something anomalous to do with the field conditions. So if there's a particularly sort of corner of the field that's particularly prone to waterlogging or that sort of thing. So effectively that's what this is, and you can see that in this instance, hopefully you can see my mouse running across the screen. In these top left examples, you can see that in the top left corner of these fields, there's a bit of variation that side of these telegraph poles. So we decided to keep the trials the other side, where we've got that nice consistency to compare between. So in terms of the first data to get stuck into, one of the things that we did was look at establishment. So plant counts across numerous plant counts down the tram lines, basically down each of the tram lines. We did those at 4 weeks post-drilling, 6 weeks post-drilling, and 8 weeks post-drilling. The idea is that then if there's any changes with time, you can see if any have a sort of lagged development in terms of emergence, and you can see that progression in time and difference between of the different treatments. Here there was no significant difference between the different treatments at any time point, 4, 6, or 8 weeks, as indicated. As hinted at by the standard deviation within this data, reasonable amount of variance, and there was no significant difference in terms of that emergence. So then what we did after we'd got that initial idea of how well the crop had developed, making sure there's no tram lines that the crop hasn't established in, we then looked at soil structure. So in some of the work packages of the Strategic Farm South, some of the trials, actually we're looking for differences in structure. And actually with time, that may be something that we would look at here, but really in this first year, it's actually about making sure that we've got comparative soil structure between the different treatments. What we don't want is to look at the soil structure in one tram line and find that it's actually really compacted, because that's obviously going to affect the results that you get in terms of that root development and some of those root metrics we'll look at later on. And the good news was that for this first year, where we look at soil structure, did VES assessments, so your standard looking at the soil structure, looking at aggregate stability, that sort of thing. It was consistent across all tram lines. In terms of here where you get what appears to be a larger aggregate, David's soil is quite flinty, so actually generally those are flints. So in the untreated, for example, most of those larger objects are actually flints rather than sort of big consolidated aggregates. And it had really good VES scores. David's soil is generally really nicely structured. It's a sort of Hampshire chalk Andover series, Andover and Coombe series. So yeah, it was really nicely structured, sort of silty loam. So once we've done those VES scores, we also then took some broad spectrum soil samples. The idea was that we could then compile all of this together into what we call a soil health scorecard. So the soil health scorecard was developed as part of a project that was sort of coordinated by HDB, involved numerous stakeholders basically and numerous different organisations providing input. Soil health scorecard, you may well have heard of them, trying to sort of publicise them fairly broadly. Basically ties in physical features, chemical features and biological features of soil, provides a traffic light system where green is things which you don't need to monitor so frequently. They're within a range that's probably acceptable and where still want to monitor it, but you don't have to do so quite so intensively. Amber is where something you probably need to investigate. It's not an urgent concern, but you need to work out what's going on there. And red is a feature that you immediately need to investigate, work out whether that's where eventually. So in terms of the soil health scorecards that we compiled for this trial, effectively there are very few differences present. Most of the differences that did appear to sort of be present were actually just because they were the value was narrowly either side of a threshold. For example, the earthworms here, if you look at that column of the sort of summary page of the scorecard on the right there. For the eco-worm application, on average, there were slightly fewer earthworms found, but the threshold for it becoming green is 8. So yeah, it's only narrowly beneath, and it's important to look at that broader context. Generally speaking, all of the values were pretty good. The pH is slightly high, but then as I say, it's a chalk soil. It's an innate characteristic of the soil, really. There's not a huge amount done about that. It's just something to sort of consider really when you're looking at the management of the site. And those other features, so K, again, it's a bit lower in the eco-worm LCBF boost, but it's not that much lower than the other tram lines. It's just that other side of the threshold. And in terms of the CO2 burst, you'd see that red colouring and you'd think, okay, that's something I really need to investigate. But given that the other features like the organic matter percentage are actually pretty good, it's a feature that's important to keep an eye on, but it's not something that would force you to change your management practices particularly. So generally very good and fairly consistent, meaning that when we then come to look at some of those other root metric data, you would expect that they're not sort of overtly influenced by any of these features in the scorecard. So we also looked at soil mineral nitrogen, how well the different plants are capturing that nitrogen. So in this first year of the project, we only looked at soil mineral nitrogen. In further years, we're also going to do tissue testing for nitrogen, just so that we can sort of tie that off a bit more neatly and say with a bit more confidence where that nitrogen is going in terms of differentially. So actually, we did see some differences here. So in November, we noticed that soil mineral nitrogen was greatest in the eco-worm and LCBF boost combined treatment. And then by March, it was actually greater in the eco-worm only and eco-worm LCBF boost combined. There's a lot of variance within the data, so it's difficult to say sort of robustly that you can draw any trends from this. Again, it comes with the caveat that it's the first year of the data, and really it's going to be about building that story over time and seeing how trends develop between seasons. Then we also sent off some samples for soil microbial analysis. So this was looking at total bacteria, total fungi, the ratio between the two. And we did that in both December and in March as well. It's important to say that we also did look at active bacteria and active fungi, but in terms of displaying the data matched up fairly linearly to the total. So in terms of displaying it, it doesn't show much that's any different. There's some anomalous data, which was part of the reason for repeating this sampling actually in March. And again, it's another part of that building the data set over time. So actually, when we look at the eco-worm and LCBF boost data, both in terms of the total bacteria and the total and the ratio between them, obviously as influenced by the total bacteria, for December, actually that value is very low. So that 140 figure that you see for eco-worm and LCBF boost combined, that total bacteria value was anomalously low. We're not quite sure what took place there, which is part of the reason why we then did that repeat test in March. And actually, by the time we came back in March and looked at it again, the values looked sort of that anomalous value was effectively corrected for. So again, in future years, it's going to be a bit of a case of just keeping an eye out for where those differences occur. In terms of the value of this data, so effectively total fungi, total bacteria. In general terms, you would expect bacterial populations to be more stable. Fungi are generally more influenced by management decisions. If you do sort of extensive cultivations of a field, you would expect your fungal community to be more affected than your bacterial community effectively. those fungal hyphal networks are going to be disrupted by cultivation and that sort of thing. So your fungi, in terms of having a higher total fungi number, you would expect that to increase where actually you've had that soil that's been intact and that population is able to develop. There's other features that influence it as well in terms of the carbon to nitrogen ratio of the material that's applied, but that's the sort of general idea of it. In terms of the ratio between them, obviously they're both outside of the guideline figures. It's important to note that here, because it's on that high pH soil, actually where previous studies have looked at how pH affects that ratio between the two, bacteria are far more affected by pH, generally speaking. So you'll generally see an increase in bacterial populations with pH. So the higher the pH, greater the bacterial population. Whereas with fungi, they're a bit more tolerant to that broader range of pHs, which might be the reason why that ratio is a bit thrown out for some of these relative to the guideline values. This actually is I'll also mention on this slide here that part of the logic how we're running the strategic farm is that all of the assessments that we're doing are all available at commercial labs. There's nothing that's going off to specialist labs for analysis. It's all things that could feasibly be done on farms. So this analysis, for example, was performed by Soil Biolab in the UK commercial lab. And the idea is that not only is it look at some interesting features and we can test out some interesting things, but it's equally about actually giving farmers the tools that they can use on their own farms to then go and investigate these things. So maybe you're thinking about introducing these products, you could quite easily replicate this exact trial on your own farm to look at these things on your own soil, on your own farm, in your own sort of weather climate and that sort of thing. So then we get into some of the sort of plant metrics. So then in December, we looked at above and below ground dry mass. Again, there was quite a lot of variance here, no significant differences in terms of the below ground or the above ground biomass, fairly similar values. And you can see them essentially vary either side of the untreated, actually. So no significant differences there. This was effectively, we took a quadrat, we placed it in numerous positions down the tram lines, extracted all of the plants present within that quadrat, cut them halfway where they sort of divide into above and below ground material, and then weighed them effectively. Some of the other root metrics that we look at. So the idea being for this was that it gives you an idea of how those roots develop over time and whether any of these products provide that boost to root development that will provide them with a bit of extra resilience in terms of weather conditions and that sort of thing, and also in terms of enhancing the nutrient uptake of plants with more developed root systems. Again, we didn't see significant differences in total primary root length or longest root length. So total primary root length, again, sampled a load of individual plants from down the tram lines, effectively looked at the total primary average total primary root length of each plant. So we ignored the sort of smaller secondary roots and mainly focused on those primary ones for those. And it's effectively the cumulative per plant total root length. Longest root length, it's exactly what it sounds like. We look at what the actual longest individual root is from each of those plants and take an average. The averages were very similar and yeah, no significant differences in those either. In terms of the root dry mass with depth, so then we again sampled some plants at two different depths. So this was at sort of growth stage 32, 33. Looked at 0 to 20 centimeter depth and a 20 to 40 centimeter depth. David being on that chalk soil, the chalk actually starts at sort of a 45, 50 centimeter depth across most of his fields. And here, no significant differences again. That's neither cumulatively or at sort of the depths independently. So there was no differences in those root dry masses at that point either. We also then sent off samples for mycorrhizal root colonization. These again, commercial lab, went to Soil Biolab as well. So again, it's something that you could, it's a relatively, to the other analyses, expensive bit of analysis, but you could feasibly do it on your own farms as a sort of commercial lab that provides these things. Basically, mycorrhizal root colonization, arbuscular mycorrhizal fungi effectively can develop a symbiotic relationship with plants and they essentially go out and scavenge nutrients to provide them to the plant. So it gives an indication of how well that relationship's working in the. So we did see some significant differences here. So we saw actually greater mycorrhizal colonization observed in the untreated plants versus the EcoWorm or the EcoWorm LCBF boost. Given that it was the untreated in particular that had that significantly greater mycorrhizal colonization, I would say you definitely couldn't say with any sort of robustness that that's actually a feature of not treating it. It's most likely that it's just sort of a feature of the natural innate variance of this characteristic of the plants. But again, it's something that we'll look at year on year, and there might be those clearer trends that develop with time. And then, in terms of that feature that actually is probably relatively near the top of most people's agendas, what did the yield look like in terms of these fields, in terms of these treatments? So no significant difference in average yield between treatments. The yield was near identical between all of them. Decent 10 tonne per hectare yields, and there was also no significant differences in terms of the protein content or the spec weight either. All of them are very similar between the treatments. And I should also mention that here the tram lines are actually harvested separately in order to prevent sort of risks of contamination. So basically trimming down a tram line with not the full header width, and then trimming down the next one as well, just to get that cleaner data. So in terms of this, effectively the take-home messages from year one, as I sort of keep emphasizing, soils are variable and long time periods can be necessary for some of these trends to be identified. This is obviously almost like a baselining year, really. In terms of the future years of this work, actually we're looking for cumulative trends. So these tram lines, that field's currently under a cover crop, and we'll go into a spring crop. I believe it's spring barley, but David can correct me off the top of my head. And effectively, it's going to be that cumulative examination. So those same products are going to be applied down the same tram lines to see whether there's an effect that develops over time, basically. As I've said, it's important really to examine whether such products will have the desired effect on your soil within your system. The chalk soil does have some distinct features. It does have that higher pH from that big stock of calcium carbonate taps with 50 centimeters under the soil. And what is that disappeared? I don't know, it's kicked me off that slide there. There we go, hopefully that's put me back on there. So it does have that some of those distinct features of the soil. So it's important to look at whether or not these are the right fit for your own farm. As I say though, the benefit of how we've run this trial is that the idea is that we've not done anything that you couldn't feasibly do yourselves, maybe with the exception of the mycorrhizal fungi where that's a bit more expensive. But otherwise, you could feasibly run this on your own farms as a tramline trial and do these sorts of investigations yourselves into how it fits within your own system. And as I say, over coming seasons might be possible to work out whether or not there are some trends that develop more so than this first year. Yeah, and I think that broadly sort of concludes this presentation really. I'll just briefly say that in terms of the plans for next year, as I say, same tram lines, same applications, cover crop going into a spring crop, but otherwise all of the assessments are remaining the same, so we can build up that time resolved data set. And also we'll also be incorporating that tissue testing for nitrogen, just to add that extra little layer of detail there.
Great stuff. Thanks, Callum. It seems to me that the EcoBoost, is it designed to enhance a soil condition that's already in good fettle, or is that one of the things that's letting it down, perhaps?
Yeah, I mean, so I would say all of David's, so on David's farm, because it's on that Hampshire chalk soil, it's all of the fields are very well structured in terms of soil structure and in terms of most of the data that pops back in terms of chemical data and biological data. All of the values are reasonable for what you would expect for a sort of chalk soil. So in terms of David's management of it, I'm not just saying this because he's also on this webinar, but they are very well managed. To some extent, that's possibly true. So for the eco worm, the vermicompost, effectively the idea is that it's broken down organic material, so it's relatively nutrient dense. Effectively, the earthworms that have broken down all of that material, the idea is that there's a more concentrated more concentrated nutrient dense product that you're applying versus just applying the actual organic material to the soil. So you would, if sort of working in this, again, it's the first year, so it's possible that it might be necessary to have a bit of a cumulative effect. But if you were expecting to see significant differences, you would expect to see them in some of those sort of bacterial and fungal values in terms of the amount of organic material available and that sort of thing for turning over within the system.
Yeah, great. We've got a quick question coming for you, Callum. Do you know how seed treatments interact with soil microbe numbers?
How seed treatments interact with soil microbial populations, was that?
Yeah, soil microbe numbers.
Yeah. Yeah, so definitely, I can certainly speak a bit on this. So there are deleterious effects. Quite often you see it with many of the products that you'd applied. So you also see it's also a consideration if you do if you have a drill that also applies fertilizer at the same time. Then also you get sort of microbial dead zones, effectively they're called, and you also can get this from some seed treatments. Seed treatments, particularly obviously the anti-fungal disease development ones, are literally designed to prevent fungal diseases, and you can also get localized deleterious effects near to seeds. The jury's a bit out onto what distance that takes place relative to where the seed is, if you see what I mean. So, or fertilizer pellet alternatively. So in terms of that kind of distance effect, I think there's some research that suggests it's quite a large distance, and some which suggests that it's a very, very small distance around that sort of circumference around where you've applied that product, where you get these dead zones. But effectively, yeah, it can reduce microbial populations. It can have a harmful effect on them.
Yeah, and a quick question here: were LCBF Boost and EcoWorm products applied more than once throughout the season, or were they just used as a one-off treatment?
So that is a very good question. David can probably contribute to this with a few more details about the specific timings of them. EcoWorm was applied at drilling, and then the LCBF Boost was applied at drilling, and then additionally throughout the season at other time points as well. I believe in spring, was it, David?
It wasn't applied on that field. We apply LCBF Boost with our liquid fertilizer on all the other fields, but for the sake of that trial, it only had the LCBF Boost with the drill.
Yeah.
Okay, great stuff. And David, how's probably easy news really, but how's the drilling season gone for you this autumn? And is it all no-till for you? And what was your thoughts on the trial so far?
Yeah, all gone in really well. Even cover crops for the cover crop trials, we drilled them and that's very dry, but they really scampered on in the last few weeks, so they look really good. I think if I can just give a little bit of background, Harry, quickly about why we've done this particular trial on establishment. When we first went to no-till with a disc drill in 2015, we saw very lazy crops because we weren't applying anything and running from a conventional no-till system into a conventional min-till system into no-till. We were seeing a detriment in how the crops were getting away. So we started looking at, we got a derogation from the Environment Agency to use DAP in the autumn, which made a massive difference. But doing tests as we went through, we were finding that we were depleting, especially mycorrhizal fungi, by using DAP at that time. So we ended up weaning ourselves off of the DAP, and really this trial has evolved because we wanted to know what to do next. And if us do nothing, great. If there are other things that we can help to enhance the establishment, then that's what we're looking for, really.
Yep, great stuff, super job. And just one more question that's come in. How do you think a cover crop, farmyard manure, organic manure input regime would stack up against the current products that you were looking at?
Well, because at that point we're looking at a winter crop, we wouldn't be able to get a catch crop in quick enough to actually do a lot of good within that. So we will see this year where we have put a cover crop across that field, and then we'll have the products going in the spring as to whether that makes any difference at all. It's difficult to run a trial across a lot of years when we're going to have a lot of different crops in there, so there's going to be a bit of variation within that, and that field is actually down for spring wheat next year, so we'll see what happens with that. But cover crops have an absolute role to play, but obviously not within or before a winter crop.
Yeah, I see. Okay, right. Well, on that, we'll move on. Thank you, Callum. Thank you, David. We'll skip all the way to Scotland and Fiona Burnett, and hear about the trials work that are happening at Balbirnie Farms.
Thanks, Harry. I'll just start sharing my screen now, so I'd be glad of a thumbs up if all goes well.
There you go, spot on.
Perfect, thanks, Harry. Yes, so talking about the spring barley establishment results last year. So spring barley obviously being a very key crop on the farm and in Scotland, where establishment is often that balance of going early to optimise potential, but the danger of going too early and suffering from establishment problems. So really interested in how we can judge the optimum sow date on the farm. Just a reminder where the Balbirnie farm is. So you see it marked there in the middle of Fife, just up from Glenrothes. And there's a map of the farm there. And some of the farms, we're in year 2 of the strategic farm in Scotland. So revisiting some of the fields that you see on the maps. And again, as Callum was highlighting there, the potential of being able to go back and build data year on year is really one of the strengths of the strategic programme. So we had 4 areas of work this year. 2 were looking at wheat and nitrogen and nutrition, and later webinars will deal with those. But we had 2 linked work packages looking at spring barley establishment. So what we did was overlay the timing, the drilling dates with different cover crop management regimes. And really, you know, in this programme and in others on the farm, seeking to investigate how cover crop practices link with soil health and link to plant health. And then we were also looking at other measurements, so it goes beyond just the crop and the yield. So we were looking at benefits to biodiversity as well, and they'll be covered in a webinar next week. So thinking about the design for the 2 direct drilled spring barley trials that we had on the farm this year, it was basically large plots, so we could really look at the management effects. We had 2 reps in each field of each treatment. So you see there the 2 fields we were working in, Tile Park and Tanks Wilson, which we were in the year before. So again, we begin to build data on the individual fields. And the cover crop treatments, the management options were essentially no cover crop, spraying the cover crop off just before the spring barley was drilled, or spraying it off just after it was drilled. So essentially direct drilling the spring barley into the cover crop, which was essentially a mix of forage rye, peas and beans. And then we had a drill date that was timed to be more or less local standard practice, and then aiming to be 7 or 10 days later than that, and again 7 or 10 days later than that. So we had almost a month between the drilling dates as they went into the field. It's always ambitious in Scotland to try a drilling date trial, so that was a great result just from the start. And then broadly, we could group the assessments into crop establishment, disease, yield counts. We did soil health measures, and then we did biodiversity assessments. Interested in Callum's comments on how to place a trial in a field. On the scale of trial we were working on and the scale of the fields, you can see that we were occupying, the bulk of the field. This is how we laid the trial out in Tile Park. So you see, essentially, we have drilling date 1 at the top there, but spanning the tram lines for easy management, drill date 2, and then drill date 3 at the bottom with the replicates of the cover crop options interspersed there. And again, in Tank Wilson Field, we're occupying almost the whole field. A similar design, except that you see that the drill dates are in one long, narrow strip along the field. But again, the early one at the top, second and last. So again, Callum's done a nice introduction to VEST scores, but we looked at the VEST scores in the fields. And again, just probing whether the cover crop made any difference. So we're looking at March VEST scores here. And when you compare the scale there, you see that Tile Park it's okay, but it's sitting in that middle of the moderate zone, so somewhere between a 3 and a 2. Tank Wilson is slightly better, so it's tending just towards that good score. We see the spring barley where the cover crop was retained, just creeping into that good zone, but you also see the variability there. So these are trends rather than definite findings. But interesting that Tank Wilson score had improved on the VEST score from the previous year where it was in winter wheat. We looked at penetrometer resistance. So again, there's a traffic light colour that I've used there, and you see that Tile Park is kind of sitting in that orange zone, firm, partly compact, but it's closer to optimal. Tank Wilson is tending to be a little bit firmer and partly compacted. And then obviously water content, very crucial to spring barley establishment. So we looked at the gravimetric water content in the fields, and you see there the trend over time. Actually, at drilling date, they're really very, very similar. Tile Park starts a little wetter in February, but really by the time we're into March and drilling for that earliest drilling time, which was the 25th of March, there's really nothing between them. They're sitting around 26, 27. Potentially Tank Wilson slightly drier at that early timing, but hard to say. It's maybe worth saying that the seed used is untreated and tested at 27% microdochium. So the potential of establishment problems arising from that microdochium in a cold, wet start might have been significant in a different season. As it was a fairly dry start to the season. And then interestingly, we start to look at things like soil nitrates. If you look at the May figures here, obviously when fertilizer had been applied, you do see a hint, again, the variability in the data is clear, but in that orange bar, which is the spring barley drilled into the standing cover crop, so the cover crop retained for longer, you see slightly lower nitrate levels, which you might expect if the cover crop is using it or locking some of that up. A similar result when we look at ammonium levels. Fair to say it's much more evident in Tile Park. We don't see the same trend in Tank Wilson where there were no significant differences there. But there is a bit of a trend there to lower ammonia levels where the cover crop is present and slightly lower again where the cover crop is retained for slightly longer in that orange bar. And a trend here, again, the variability of the data shines through. But again, if you look at the May figures, there's that slight dip in Tile Park when we come to that retained cover crop in orange. So just that hint that the nutrient levels are slightly different in those cover plot treatments. So then if we move on to the influence of drilling date on establishment, just some images there which show you emergence in Tile Park and Tank Wilson towards the end of May. So that's 2 months after drilling for the earliest drill date, which you see there on the left. And then for the right, we're looking about 5 weeks after drilling. And again, you see how strongly established that early drilling time appears. If we start to convert that into numbers, we can see we do see an influence of cover crop, but we see more strongly that influence of drill dates. So the coding there, the cover crop, nothing at the bottom, sprayed off before drilling as the B, and then sprayed off after drilling is the A at the top of the chart there. And really, drill date 1 at this early assessment timing is leading in both fields. But the influence of the cover crop differs most for the later drilling dates. So it's as if that later drilling, the more challenging it becomes, the more challenging other conditions become. So you see that grey bar trailing behind where the cover crop is retained for longest. And that's probably more evident in Tile Park there. If we look at plant counts per metre squared, they're much more similar for drilling dates 1 and 2, and that slight penalty for drill date 3, so that grey bar dropping behind for every scenario with the different cover crop options. But again, if you look at Tile Park and that influence of the retained cover crop, it becomes more evident that the drill date 3 is the one that's really tailing behind in that option. So just flicking forward to a slightly later drill date here. So we're at the end of July now and looking at plant counts. And by this timing, it's interesting how the crop has begun to compensate. And there's still a penalty for the later drill date. So for that drill date, well into April. But drill date 2 in early April is actually beginning to pull ahead in terms of plant counts. And in Tank Wilson, the influence of the cover crop options in terms of plant counts has pretty much vanished. It's still evident in Tile Park though. So again, we see that drill date 3 is perhaps struggling more in the cover crop options. And again, that trend to the later retention, the direct drilling into the cover crop in A being the one that is potentially problematic. And then obviously disease was a very important measure as we go through the strategic farm programme, really seeking that link between, good soil health, strong plants and better disease levels. We've been blessed in our 2 years with 2 quite low disease seasons. So last year, because it was very dry, the spring barley crop was not particularly at risk, but we picked up some Rhynchosporium towards the very end of the season. Disease levels are slightly odd in that because the early drill date had started to senesce, it appears that the Rhyncho on leaf 3 that we're looking at is slightly lower. There is a trend there that the later drill date in Tank Wilson certainly has less disease, and that kind of conforms with perceived wisdom that later drilling helps to diminish risk. But the variability there is very high, and like I say, because the early drilling had started to senesce, it's not hugely meaningful when it comes to saying anything about the early drilling date levels. So looking at yield, and again, because we were occupying large portions of the field, you can see here just the overlay of the plots onto off the combine yield maps, which is how we generated the yield data. And as ever on farm trials, you see the problems in terms of yield mapping around the edge of the field, and a hint to a lower yielding zone at the bottom of the field there, which may have unfairly affected drill date 3. But you do begin to see, even visually there, you can pick out how the plots were looking. And that's how the yield map looked in the Tank Wilson field. So again, it's always interesting looking at yield maps. You see a hint of a left-right split, so hinting at probably an old field margin there, with the right-hand portion of the field still yielding slightly lower. But a couple of low-yielding treatments you can pick out fairly visually just from that yield map. But squinting at pictures is not what we're about. So here's a graph. And yeah, the drill date one is leading in Tile Park. So I think what we're beginning to see here, again, is probably self-evident, but that actually even on the same farm, it's very much down to the individual field conditions, and within that, the treatments within the field. So yeah, in Tile Park there, you see that drill date 1 leads for the 0 cover crop treatment and for the later retention of the cover crop, and it's much more evenly matched for the cover crop retained but sprayed off just before drilling. And in Tank Wilson field, drill date 2, interestingly, is tending to be optimal, and actually drill date 3 is optimal in that no cover crop option. So that again is just, it didn't particularly come through as we went through the season, and yet that has been the optimal in terms of yield option for that particular field. And I've just put that into numbers as well. Sometimes we can go graph blind. So averages can sometimes tell you nothing, but what I wanted to say here was if we look at Tile Park and you look at the average for the early drilling date, you see that it's sitting there at about 6.32. And in fact, for all the cover crop options, the high yields fall in that option. And again, if you look at the A, B and 0 lines, it's, you know, that the highest average is 6.7 for the no cover crop option. And that's true across most of the data that you're looking at. Tank Wilson just shows how the story can slightly twist. So yeah, the average for drilling date 2 appears best. And again, it's that no cover crop option that is appearing to be the highest yielding. But that late drilling date that popped out for that combination of, you know, late drill date 3 is actually the highest yielding treatment in the field. So just those micro details that make all the difference to how well a crop will established. So just a few key findings and observations from what we've done this year. Clearly, sow date was a very significant driver of spring barley establishment during the season. And we saw there, even on the same farm, how optimum sow dates differed between the two fields. And in Tile Park, drill date 1 was optimal. Tank Wilson, drill date 2 was the optimal overall, but with that slight option that for drill date 3, where there was no cover crop, it actually yielded slightly higher. Tank Wilson, we know, had a slightly higher VEST score, but it was more compacted. It was potentially slightly drier at the start. Late drilling definitely carried a penalty in most scenarios in both fields, but it was much less pronounced in Tank Wilson. Tile Park was by far the higher yielding field. And the yield penalty was more pronounced where the spring barley was direct drilled into the cover crop. So that was really evident in Tile Park. And that's a really interesting finding. And there's a number of reasons why that might be so. So potentially, Tile Park was the field where we saw some of that nutrient lockup, where the cover crops were retained for longer. Or was it just something more physical? So was it more challenging to establish in a direct drilled scenario? We know we had fewer plants and less cover early on for those scenarios. So again, these are things that by repeating the trial in the coming year, we hope to unpick a little bit more, but it really demonstrates how variable an optimum can be even within the same field. And as I say, we're repeating that in 2023. David's blessed us by adding sheep to the trial next year. So that grazing effect will be something that we'll look at as well. And then, like I say, it's not all about yield. So we'll be looking at some of the biodiversity benefits that we measured in the course of the season in the webinar next Wednesday. So that's a spoiler slash trailer if you wish one, Harry. And that's me, finished my slides, thanks.
Great stuff, Fiona. Thank you very much. Couple of questions coming in. One from me, just quickly. The nutrition for all trials was the same across all of the plots?
Yes, I'll let David confirm that, but the only difference was the cover crop treatments and the drilling dates, obviously. Is that true, David?
Yeah, that's all the pursuing treatments were identical, other than the cover crops and the dates, obviously, as Fiona said.
Yeah. Okay. A couple of questions coming in. If you've got, or to the audience, that if you've got any burning questions, keep them coming in. We've got one here for Fiona. What's the difference between gravimetric and volumetric water content?
The gravimetric is a percentage of the water content rather than the volume, but I don't know off the top of my head how you convert one to the other, but they're linked.
Okay. Okay. And was there any consideration to subsoiling based on the penetrometer results?
I'll let you take that one, David.
David.
No, but perhaps I need to recalibrate my foot and my spade, because when we dug holes in the field, there didn't appear to be any compaction. Interestingly, we did do a bit of low disturbance subsoiling on the end rigs just before the fields were sown, and it felt good to do and it looked good to do, but the yield map, which it was a waste of time. We didn't get any more yield as a result of it, which I was surprised and disappointed at.
Yeah, okay. And was there any difference in grain nitrogen? Did you test that?
We didn't measure grain nitrogen in this trial.
Yeah. Another question.
All made malting, so it was still fine.
Okay. Where are we? Another question coming in. Have you considered earlier destruction of the covers?
David?
Yes. We're quite we're a sandy loam here, so we're not into the realms of these people that have clay soils where the worry is that you're having too much moisture in the surface. We did especially pick a convenient time when the spray, when we can get on with the sprayer and take it out. I suppose that was what the reason behind the trial was. Does it make a difference? And it would appear on year one of information, it does make a difference. And that's partly the introduction of the sheep, which we're doing on the rest of the farm, is partly to try and compensate for the fertility a bit and take some of the fertility that's wrapped up in the cover crop and put it back into a form that the crop can get a hold of. And one of the fields, the Tank Wilson's Marsh, we had an awful slug problem. Not for establishment, it was fine at establishment, but once the crop came through, the slugs just seemed to hammer every leaf that came, which was not what we usually see. So whether we can try and create a slightly firmed up surface with some sheep to try and put the slugs off a bit and give ourselves a bit of a chance, I don't know.
Be really interesting, yeah.
And had the slugs survived through with the cover crop? Would that have been had an impact?
We've got some quite nice data, which I seem to be plugging next week's webinar quite solidly, but okay. Slugs and some of the beneficials were, particularly what we were looking at, Harry.
Yeah. Okay, great stuff. Well, that's the reason to tune in for next week. One other question before we go on to David Blacker. Two other questions in actual fact. Was there any impact on gross margin?
David, you've got the benchmark data, so.
Yes, it was whilst the results that Fiona looked at weren't significant in terms of statistical significance, the impact on the margin, particularly with the grain prices where they are now, was quite a bit. but not hugely variable. Yeah, we had similar yields on other parts of the farm, and we had other fields that did better under same circumstances. It was just a hugely challenging and very dry season for us in Fife. So I think that's basically what dictated what happened this year.
Yeah. Okay. And then one final question from the floor. Does post-drilling glyphosate give a cleaner establishment and reduce the need for post-M herbicides?
It could do. We have done it in the past. On one field in the past, we got away with no other herbicide bar the glyphosate before the crop came through. Really for us, pre-Ms are about annual meadowgrass. If we lose control of that, obviously we've nothing else in spring barley to deal with it. So we did apply a pre-M as well, because we just don't want the meadowgrass getting a hold. Although we do tend to find that when the soil structure comes good under the direct drilling regime, that we tend to see meadowgrass just confined to areas of compaction. So it's quite a good indicator, actually, just by walking across the fields.
Yep, great stuff. And penetrometer-wise, David, it's a spade rather than a prong with a gauge on. You just go out with a spade.
I do, but I don't know. Maybe I ought to get a penetrometer, and I can be a bit more objective about the information.
But
We'll share.
Does the spade pick up as much? Once you get used to using the spade and you go out and you get a feel for it, it can tell you as much. And you've got an opportunity to look at soil health and fungal activity as well.
Yeah, you can see more with a spade. And we've got a lot of stones in the soil too. So every second time you put the spade in the ground, you hit a stone. So it takes a while.
Yeah, fair enough. And drilling wise, David, that went pretty well for you in Scotland. All done, all up to date.
Yes, it's fantastic. I've never seen things looking so good. You've got to go a long way to find a bald spot in a field around here this year, but I think that's probably the same can be said in most of the country. When nature gives you a helping hand, the job's really quite straightforward.
Yeah, great. Callum, you've appeared. Did you have something to add?
Yeah, I was just going to add on the penetrometer that actually with particularly stony soil, so one of the sort of thought processes behind not incorporating it within David's farm, that on very stony soils, which David did, is probably 40% flints by volume, probably on that top soil surface. Is that actually where you use penetrometers on stony soils, you can get really unusual numbers. Where you get resistance provided by stones at the surface and also underneath. If you imagine that it's effectively applying a pressure to the surface, you get sort of shears of soil moving around stones buried underneath the soil. So you can get sort of funny values basically reported back by that soil moving around stones under the soil and also at the surface as well. So it's just something to consider, really, that sometimes a fork and a spade can help you out in those sorts of situations. Yeah,
good stuff. Yeah, just the job. Okay, well, thank you, David Aglen, Fiona, and Callum. We've got, as you know, we've got David Blacker on the line as well. Hopefully, he'll come on in a minute. We haven't got a suite of slides from David or from the ADAS team that are doing the work. But we've got a kind of a quick synopsis of what work is going on at David Blacker's, and it's quite different soil type. Though we're not going down the regen route just yet, and we're not in the no-till world just yet either. But welcome, David. I thought I'll just share some of the work that we've got. We've got 4 themes of work really. I'll start with the 4th one to begin with. Drainage trial. The objective is to investigate the impact of drainage approaches on crop performance. We're going to compare new laterals, new drainage system in the field. I'll let David describe the field in a minute, at 10 and 15 and 20 metre distances. And this is in addition to the old drainage system, the clay pipe drainage system there. And then we'll assess really the soil biology, chemistry, the physics will be tested, pH, soil organic matter, organic carbon, soil texture from the soil surface to a depth of 15 centimeters, penetrometer again, earthworms and bacterial fungi ratio. We'll test the drainage water over winter to see what comes out of the mains at the end of the field, and then look at crop establishment and whether that can be optimized. Let's put it that way. Looking at plant counts and NDVI, routing, tissue analysis, and biomass. I said biogas now. But David, it's certainly an interesting field. If any of you have been to a monitor farm meeting at David's in the past, will have no doubt been to the field that's alongside the East Coast Mainline Railway. Those drains running yet, David?
Yes, they are running like the clappers. I don't know what it's like up and down the rest of the country, but yeah, it's got very wet very quickly up here. So yeah, I was looking at some drains yesterday. The new systems now are running so fast in one field that the old main that runs into the next field it has to go through can't cope with it. So yeah, it's nice to see. It's nice to see it moving off the field and not just sitting on top and affecting crop performance.
So okay. So it's made a difference already.
Yeah. Yeah, well, you know, I mean, so the history of that field, the one we're actually doing a trial in has been one of the biggest challenging fields I've got on farm. I've got a few more areas like that I need to address yet, but it's one of those fields where it's just the oil system just ceased to work, and it kind of left me praying for a dry autumn in order to get a crop established and into spring. And more often than not, in a Vale of York, we don't get that. So I've probably had more crop failures in that particular field than anywhere else on the farm, to the point that a lot of the time it's just been written off. The previous crop in there was a winter oilseed rape crop that going into December was kind of knee high, best crop I'd seen in there for a lot of years. And we had a -6 frost, and it killed all the rooting zone stone dead. It just froze it like an ice cube. So that's where I properly spat my dummy out and said, right, I'm having this. I'm sorting this out. I'm sick of this. But again, it's also great high silt content in there, so it's a field that does slump and cap very easily, and I think that's been exaggerated just because the water's never had anywhere to go, so it just sits. Once it's saturated, that's it. It can't go anywhere. So, frustratingly, you can establish a crop, establish it well, and then slowly watch it die a slow and waterlogged death through winter.
Yeah. I've heard you mention ducks per acre, which don't really pay any bills. So.
No, it's not good for your profit margin.
No. And you've got the machinery in yourself, so you've got your own trencher and stone trailer and so on.
Yes, so I mean the bits of draining I've done before by contractors. You quickly realise that it's a very fast way of writing checks out to get some field drainage done. And I kind of looked at the area on the farm that I had to do, which was most of it. And the speed that it was all collapsing and failing, and just thought, it would be financially a better decision just to have my own kit, and just to go out and do it when we had time. So in June, July now, early July tends to be drainage season. I've been following the fields and taking them out of production just to give me the window I need to go in there and do the drainage. When I started growing spring beans, I had this this ideology that I'd be able to drill a field through winter, through a standing cover crop before spring beans. The reality is it's never a dry field you're going to, and by the time we get drilled up, the field's too wet to get on anyway. So that never really happened. So I've reluctantly been following bits of land just to leave them there available to go at. I've done a bit through crop, but it doesn't sit that well when you've invested all the money in a crop. I've kind of taken advantage of the stewardship schemes, and I've actually got bits of fallow now in my stewardship schemes, so I can do the overwinter summer fallow option take a payment for that, and I'm also allowed to do drainage through that option. So that kind of makes it a bit more palatable. As much as I hate fallow and I hate fields with nothing on them, it makes it a bit more palatable when you're getting paid for creating that habitat.
Yeah, fair enough. And the field you've drained with the laterals trial on has got a crop in at the moment?
Yes, it has. Yeah, it's got a crop in it.
What crop?
It's got a crop of beans in it.
Okay. Yeah. Good stuff. So then let's just have a quick look at the data analysis. I know it's machines and software systems are producing an awful lot of data analysis. So the other project that we have running on David's farm is to analyze existing data. And I know you've got more than 10 years worth of field maps and data to try and crunch down, probably more than you know what to do with. Identify knowledge gaps, trends that could influence or future trials and improve yield. So cleaning data from the past 10 years here and highlighting areas for testing and trialing in coming years for the project. So a lot of data, David.
Yeah, you're right. I mean, there's more over the years. You know, I've kind of added more things into a soil test, especially in that. Yeah, we just started doing the basic for years. We just did the basic, PK mag lime like everybody did, and then we started adding organic matters into that, and then the more you get into it, we then started doing CECs, and then if you add in a few other nutrients, you can work your CEC out yourself. So we added them in, and then it was just as cheap to get a full spectrum nutrient test done because we were doing everything else pretty much anyway. So we started doing full spectrum, and then we've got some more structural kind of data like bulk density data. We've got some of that now that we've done on farm. So you start layering all these maps out on a desk and then look at your yield map and then try and work out what it is making a difference to yield. We've done the soil as a percentage silt, percentage sand, percentage clay, so we know what the soil types are. And my go-to was always to look at organic matter to see if there's a correlation to that to yield, because I'd quite like it to be that. I just can't prove it. And it's so variable that if I was almost trying to talk myself into it being organic matter, so I was putting bias into what I was looking at. So I had this idea that one winter I'd sit down and I'd put it all on a spreadsheet and tabulate it, and just take my emotive wants out of it, and just try and get some straight hardcore figures from a data analysis package. The only issue being I don't have the computer skills to do that, and I haven't had the time to do it either. So this makes a good opportunity to just to physically look at the data against yield, and is there a combination or a go-to that's actually making difference. So that's kind of what.
Yeah, and you've been using variable rate based on Yara N sensors for years as well. So have you recorded that into the system?
We've got that. Yeah, we have got that data, so we can feed that in. I'm aware the enormity of data I'm going to be throwing at ADAS will be quite horrifying from their point of view. But yeah, we've got data on pretty much everything. The only gaps really I've got is probably more on the biological side.
Yeah.
So that's what one of the benchmarking things we're going to look at. The fungal bacterial ratios, and try and build on that a little bit. We've got carbon burst data. I just want something a bit more than that, something a bit more detailed than just a carbon burst. So we're trying to fill the gaps from biological, structural, nutritional, and try and pull it all together in a completely unbiased way to see if there is anything stand out that is making a difference.
Yeah. Okay. And I'm going to try and stay on the unbiased bit here, but playing devil's advocate, if anyone who is listening in from Hutchinson's, they'd be jumping up and down saying, "Well, you're born for Omnia." and you can overlay all of your maps and come out with a wonderful answer. Is that something you've looked at?
No, I haven't. I've never looked at Omnia. Maybe Dick Neal's not been working hard enough. He maybe should get on the phone and try and twist my arm.
Special deals are acceptable. Yeah, okay, very good. We've had a question just returning to the drainage for a bit. How do you balance drainage water to remove water over the winter, but also managing soil structure and water retention to cope with more extreme drought and weather we seem to be having? So I suppose it's not having too much drainage.
No, I don't think you can have too much drainage. I think that's a bit of a fallacy. So the drainage, the drains are there to take the excess once soil's saturated. So adding extra drains in there won't make a field drier than it should be. It'll just, if anything, it'll do the opposite. It'll create you bigger windows to work in. You'll be able to travel. You'll be able to get on the field faster than you ever would. So yeah, no, that's not. I mean, my topsoil and subsoil is relatively moisture retentive, so my issues have generally been having too much, not having enough.
Yeah. I think the person asking the question needs to come to stand in the field in question, and then they'd understand.
But there is a bit of lighter land. I mean, not much, but there's a bit of lighter land at the end of that field. Hopefully we'll pick some data up off that. But that, the drainage thing was more about, we haven't done drainage work since Dick Godwin did it years ago. Yeah. And he took some of his data to America, and America's picked it up and ran with it. So if you Google flow rates through a drainage pipe or something like that, if you put that into Google, you'll get any amount of stuff from America that'll give you, flow rates in gallons per acre. Whatever from the American sites. There's loads of data there. We have nothing. We've never done anything with it since. So it's as much as what's financially viable. Do we have to go down to 10-meter centres? Is 20 adequate? And 20-meter centres was a measurement that Dick came up with, which is why we've used it as an industry for years. Is it appropriate on very heavy clay soil, or should we be narrower than that? So we're going to cost all the drainage runs out. We're going to cost each system out. We've actually left a bit of the field in there undrained, so we can try and get some financial payback data at the end of the program as well. Okay,
yeah, great stuff. The next objective really on your farm then, David, is to assess the impact of soil health and structure on plant development and performance, and to evaluate if there's any correlation between soil nutrient availability and so to basically chasing yield. So we'll be looking at monitoring crop from establishment through development into yield, and grain analysis and plant counts, NDVI, tissue analysis, rooting, disease and biomass. But that's by and large monitoring soil for yield, really, David, do you think?
Yeah, so I mean, this kind of stems from why do we get the yields we get, and can we get any better than that? Yeah, we do see a lot of variation in fields, having several different soil types, and quite often in the same field. So it's kind of a step back and a canopy management kind of assessment, really, to see are the yields we're achieving as good as we can get for that area, or can we do better? And if we can do better, what can we do to get them better? So it has definitely got a swing on yield and making sure we're maximising what we've got. I'll try and identify where we're getting it wrong, if we're getting it wrong.
Yeah. And if Roger Sylvester Bradley was on the call, then he'd be saying yen is the way. That if we get a yen analysis, then it would signpost to what needs to change. What do you think? Do you get
we are going to be feeding a bit into yen as well to extract the data, and then we'll give that data to ADAS and get them to put that through their spreadsheet.
Yeah.
I mean, yen's not something I've done regularly, but yeah, we've done it. I've done it this year on wheat and spring beans. I've done the bean yen. Hopefully we'll be a bit more organised next year and get all the information that they want.
Do you know the potential for yield via yen in your area?
Yeah, it's something quite fanatical, like 17 tons a hectare or something ridiculous. Well,
Yeah.
if that is, then I'm not even getting 2/3 of it a lot of the time. So
Yeah.
where's the other third going? Is it? I don't know. I don't know how you get that. Well, I mean, I suppose I have a different. When you sat on the combine and you're watching everything come in the front, generally the lowest yielding areas are the bits that don't have enough plants on them. There's just not enough there to go in to compensate for the lack of tillers. Interestingly enough, some of the champion wheat growers in the world, the ones who are going for the wheat trophy, the Guinness Book of Record job, they're intentionally planting very low seed rates in order to get the crop to tiller as much as possible, knowing that for every tiller they get extra plant roots. So that they're doing exactly the opposite to me. So take from that what you will, but if that's the case, I've been around long enough to see low seed rates, and I've tried low seed rates, and I wasn't that impressed with what I saw. And again, I think it comes down to this, the state of the drainage and the soil types. But my crops never seem to tiller profusely well in a wet Burley York winter. So that's not an option. I'm choosing to put a bit more seed on to start with and make sure I've got a decent plant count there from the beginning.
Yeah, and do you have an established approach to cover crops, or is it just trying something different every year?
Not really. Keep it as cheap as possible. Really, is the only constant I've got.
And the aim being green cover, or root density. What's the aim for your cover crops?
No, my aim is really restructuring soils. That's the biggest benefit I can see from a cover crop.
Yeah. Okay.
I can quickly go down a rabbit hole here if you want with cover crops.
Say it again, sorry.
I can quickly go down a rabbit hole here if you like with cover crops.
No. We're doing good for time, so let's keep it moving. And then the final work package and project that we have on your farm, David, is the soil biology and soil health to depth. And again, it's on this famous field at Overton, looking at biological, chemical soil assessments to depth into the subsoil as well as the topsoil. So for the topsoil, chemical pH, soil organic matter, organic carbon, extractable P, K, and magnesium and soil texture to a 15 centimeter depth. Then structural penetrometer resistance down to 45 centimeter depth. That will do the VES, visual soil assessment, to 30 centimeter depth. And then again, we'll move on a bit into the subsoil, pH, soil organic matter, same sort of thing, soil carbon, soil texture, and the structural soil. So any thoughts on that initially, David?
Yeah, so this really stems from, I've got so much data now, and whenever we look at soils, we tend to only look at a spade depth deep. You go out with a spade and you dig a spadeful, you don't really go any deeper than that. All the data we've got for all the nutrient stuff comes from the the top 15 centimeters, but deeper than that we don't know what's going on. We have very little subsoil data for anything, for structure, for nutrients. Obviously we're not doing the biology to depth because that'll all be in the top, but it's just to try and build another layer of information on top of what we already have, I suppose, really. I have seen with penetrometers quite regularly. I can go down and suddenly hit a pan, and we've identified before pans that are down at, we're not obviously traveling this to this depth, but yeah, I've seen pans we probably caused when we were growing sugar beet 15 years ago, down at 24, 30 inches, and don't know what to do with it. I don't know how we put it there to start with, and I certainly don't know how to take it out. one of the answers has been just to redrain the field and just cut through that pan and allow the water to go somewhere. But I know there's I suspect there's structural issues in my subsoils, and unsure really how to sort them out. What the best way of sorting them out is.
Mechanically, it's not reachable.
Not that, yeah. No, I mean, there's not all that depth. Some, the penetrometer it does pick. I don't know if it's just a natural change in soil type, because that can throw some strange results when you're hitting the same thing at the same depth on a regular basis. It strikes me it might not be mechanical caused. It could just be a natural change in soil type. So, so it's more of a benchmarking exercise, really, just to put a different layer of information into the system to work from going forward.
Yeah, 3 years of Lucerne, would that work?
Possibly.
I don't know what you do with the Lucerne, but I know it's good routing. But yeah, it's certainly something to think about again. So, well, we're coming up on 2 o'clock. I think that's everything from that we'll do with you, David. If we can bring the rest of the team in. I don't know if we've got any more questions coming in. I've got Fiona in the background handling the questions. No, there's no more questions. So that's great. That's good. That's about Bob on time. So it just leaves me really. I don't know, Fiona's gone, but it just leaves me to remind you of the next upcoming webinars. We've got next week farming and environment with Anna, tailoring crop inputs with Fiona, and the future of sustainable arable business with Isabel. And same time, same place, 12:30 till 2 in the next few Wednesdays of November. But it leaves me to say thank you, Fiona, Callum, David Miller, David Aglin, and David Blacker for your help in this webinar. And we hope to see you next time. Thank you very much.